Atmic Biotech Solutions Private Limited.
I am going to discuss about one of the application of PCR, molecular identification of organisms using polymerase chain reaction and Sanchez sequence. How we can identify an organism at molecular level. Molecular identification is more precise and accurate compared to other conventional methods. For this purpose, we have to amplify the molecular marker genes from your test organism, and you have to sequence it using Sanchez sequence. For example, in bacteria, the molecular marker gene is cystinous rRNA gene. What is the speciality of a molecular marker gene? The molecular marker genes will be conserved within a species, but it shows significant variations among species. That is the difference between normal genes as well as a molecular marker gene. Here in bacteria, the cystinous rRNA gene can be considered as a molecular marker gene because it has a certain property. Because if you take an Escherichia coli from here and if you take another Escherichia coli from an Arctic region, if you sequence that cystinous rRNA gene and after sequencing, if you blast that sequence, you will get the result as Escherichia coli. So that is the advantage of a molecular marker gene. From where you isolate that organism, that doesn't matter, but at molecular level that organism is same. Suppose you are going to isolate a E. coli and also a Pseudomonas from a soil sample and compare both. The cystinous rRNA sequence must be different. So that is the advantage of a molecular marker gene. In higher organism, more than one molecular marker genes are available. So in short, we can say that molecular markers are the foundation of molecular identification. Both mitochondrial DNA in higher organism, mitochondrial DNA, and in lower organism, ribosomal DNA, rDNA, are the species specific and can be used as molecular markers for species identification. In fungi, the molecular marker gene is 18S or ITS, and in microalgae we can amplify 18S and sequence it, and in protists ITS and ATS can be used as molecular marker gene. In plants, RBCL and MATK are the most prominent molecular marker genes. In animals, in both invertebrates and higher animals, cytochrome oxidase and cystinous, mitochondrial cystinous rRNA gene can be used as a molecular marker. This is the workflow of molecular identification of an organism. First of all, you select your organism. It can be a bacteria, plant, animal, whatever it may be. So isolate the total DNA from it. For that purpose, commercial kits are available, or you can go for conventional DNA isolation methods. Based on your organism, you have to select the appropriate DNA isolation method. So after isolating the DNA, universal primers are available for amplifying the marker genes. For example, if you are amplifying bacterial cystinous rRNA gene, universal primers will be available. The commonly used one is 27F and 1492R primer. So those primers you can get it from literature and order those primers. You can amplify that cystinous rRNA gene from the isolated DNA, then go for Sanchez sequencing. After getting the sequencing result, go for blast and identify the organism. So by following this molecular identification pathway, you can precise and accurate can identify specimen. So as an example, I will give how we can identify a bacteria by molecular identification. First of all, you have to prepare a bacterial fuel culture. So pure cultures are essential in case of bacteria, because if your starting material is a mixed one, you will get a mixed reads in Sanchez sequencing, so you can't identify the organism. So the starting material must be pure. Then go for DNA isolation. Then using that 27F and 1492R primer, you can amplify that cystinous rRNA gene. Then proceed for PCR. After PCR, you have to purify your PCR product before Sanchez sequencing. For that, you can use commercial kits. After the purification, the amplicon, PCR amplicons will be sent for sequencing. After sequencing, you will get a result as shown in the left side panel. You will get a nucleotide sequence. Go to NCBI BLAST and copy paste that sequence, then search. Then you can identify which bacterial strain you have. So this is the workflow, and this workflow is common for all the organisms. The only thing is the DNA isolation step and PCR will vary accordingly. The other steps are common. These are some of the universal primers for identification of various organisms, which is available in our lab. LCO and HCO primer, which can be used for identification of insects, mollusks. Most of the invertebrates we can identify with this primer. Then ITS 1 and 4, which is used for the identification of fungal strains. Then RBCL, Rubisco Labs subunit, which is used for identification of plants. Then bacterial identification I have already mentioned, 27F and 1492R. For algae identification, again we can use 18S. These primers are right now available with us. For those who have any queries, you can contact our Atmic office at any time.